to say good morning or good afternoon or good evening, wherever you might be sitting at this current point in time, and to welcome you today to this webinar session on science policy for biodiversity research. So the session today and the first session 2 weeks ago have been organized as a collaborative joint effort with the Global Genome Biodiversity Network, so the GGBN, and the European Reference Genome Atlas, ERGA, with the very much appreciated support of the Digital Sequence Information DSI network and the Earth BioGenome Project, the EBP. My name is Rob Waterhouse, based here at the SIB, Swiss Institute of Bioinformatics, and today representing ERGA as the current chair of our initiative in Europe that now counts more than 1,000 members. ERGA is a community, if you like, of scientists that are aiming to work together and learn from each other in order to improve the coordination and to facilitate the generation of reference quality genome assemblies for the entirety of European eukaryotic biodiversity in a distributed manner across all European partners. And so this is very clearly an ambitious and complex task that will need a really concerted effort by a great number of people. And as we get started in this endeavor, it's really becoming clear that policy for biodiversity research is a particularly complex issue where many ERGA members are seeking guidance and help to really make sure that they're following the best practices for ethical and legal sampling of species across Europe. We already 2 weeks ago learned a lot about access and benefit sharing in the last webinar. And so today we're really looking forward to learning much more from the experts on digital sequence information and DSI in science policy for biodiversity research. And so just before I hand over to Katie Barker, who will be representing GGBN today, we're going to now launch a quick poll here on Zoom to ask those of you who wish to receive a certificate of attendance to please enter just your first and last names, and that will allow us to register those present so that we can send out certificates later this week for those of you who wish to receive a certificate. So with that, I'll hand over to Katie.
Thanks, Rob. So hi, everyone. I'm Katie Barker. I'm the Senior Science Program Manager at the Smithsonian's National Museum of Natural History, and I run the Secretariat Office for the Global Genome Biodiversity Network. So just a little background on GGBN, for those of you who haven't heard it before. GGBN was founded in 2011 to fill a critical gap in the existing research infrastructure landscape. It consists of DNA and tissue collections at natural history museums, botanic gardens, universities, culture collections, environmental sample collections, gene banks, seed banks, and zoological gardens, so a lot. Basically everything that's non-human. These collections provide long-term storage and public access to material used for molecular research of all kinds. The GGBN data portal and the data standard have been developed as complementary tools for other infrastructures such as GBIF and INSDC by enabling standardized access to its member biorepositories and establishing cross-links between underlying voucher specimens, sequence data, and publications. So today we have about 6.6 million records available through our data portal from 39 biobank providers. And we have about 112 members at this moment, and many of them are working towards online discoverability. So together with CTAF, Spinach, and Synthesis Plus, GGBN provides best practices for biorepositories on implementation of Nagoya and ABS regulations. And in addition to that, GGBN also provides a community document library for biobanking knowledge exchange. So for our session 2 webinar topic today, the digital sequence information and convention on biological diversity, I'm happy to introduce Dr. Amber Hartman-Scholz, who is a member of the GGBN Executive Committee, and she also chairs the GGBN Task Force on policies related to management and stewardship of genomic samples. Amber is a microbiologist and head of the Science Policy and Internationalization Department at the Leibniz Institute DSMZ in Braunschweig, Germany. She leads projects on access and benefit sharing, digital sequence information, pioneered the DSMZ's registered collection status under the EU Nagoya Protocol Regulation 511/2014, and co-founded the DSI Scientific Network and German Nagoya Protocol Hub. She held previous science policy posts in the White House, Office of Science and Technology Policy, as Executive Director to the President's Council of Advisors on Science and Technology, the National Cancer Institute as a policy advisor, and a science fellow to the California State Senate Environmental Quality Committee. She received her PhD in biology from the Johns Hopkins University in 2009. So now I will happily hand over to Amber for her presentation on digital sequence information and the Convention on Biological Diversity.
Oh no, of course it stopped right in that second. Am I still here? Am I back? It like dropped in and out. Amazing.
You're back, yes, but the
Okay,
slide's on.
I have no idea what Zoom is. It's been a real IT week for me. Katie, thank you for the kind introduction, and thank you, Rob, for the great overview of the context of what we're talking about today. Katie, you should be congratulated for saying Braunschweig with a beautiful accent-free expression. That's not an easy one for the tongue. So as Katie indicated, we're going to take a deep dive into the second half of this series, and I titled the talk for today, an effort to find compromise on benefit sharing and digital sequence information coming from a scientific perspective. The organizers asked me to spend about 45 minutes talking to you today, and we have exactly the same amount of time or even more for questions and discussion. And I very much look forward to that and hope that I can kind of provide you a history and the context of the important policy decisions that are ahead of us. So with that, I am going to start at the really big picture of multilateral environmental agreements, or MEAs. And you can see by the title of these MEAs that almost all of them, probably all of them actually, requires science. And for me, science policy is a two-way street. It's the use of scientific information to make ideally better policy. And it's also implementing the policy decisions that have been made into scientific and, more broadly societal practice. So last week, or 2 weeks ago from Scarlett, you heard more that latter of taking policy decisions and implementing them into the scientific practice. And you already remember, hopefully, that we've zoomed in here to the Rio Earth Summit treaties. There were 3 that were opened in 1992 in Rio. In Brazil, the UN Convention to Combat Desertification, the 1992 Framework Convention on Climate Change. So that is like the Paris Accords or the Dubai meeting recently. And then for our specific interest today, the third is the Convention on Biological Diversity. And for today's talk, we are not talking specifically, at least not yet, about implementing policy and doing compliance following the laws that have been written. We're talking about how the scientific perspective can be implemented and used for policy-making decisions, recognizing that it will come back full circle ultimately, and in fact, maybe sooner than we even know. So just a very, very brief reminder, the objectives of the Convention on Biological Diversity are actually quite clear. So the conservation of biological diversity, the sustainable use of its components, and the fair and equitable sharing of the benefits. We're zoomed in on objective 3 of the CBD, and the overarching principle is just like with oil or gas or minerals, you cannot go into a country's sovereign state's borders and take and use its biodiversity without asking permission. That's the sort of fundamental starting point from our conversation, and I think Scarlett 2 weeks ago covered this beautifully. So the big picture is that from 1992 to about 2016, the CBD, as well as other United Nations fora, focused very much their policy-making decisions on access to biological samples, which they termed genetic resources. For us as biologists, you can understand it as basically every kinds of physical sample that is non-human. And containing molecules of heredity, DNA or RNA. But in 2016, that paradigm shifted. Now, before I take you in, I just want into how the paradigm shifted. I did want to just remind, because I'm sure there are people here that weren't there for Scarlett's webinar, of the major take-home messages so that we're all on the same page. So number 1, Scarlett told us that fair and equitable benefit sharing is a key principle in international environmental law that is uncontested. That is the way that it's going to be as long as there's the United Nations. Number two, that access and benefit sharing laws, in particular under the CBD and then later under its Nagoya Protocol, are made at the national level, and they involve both rules about access and rules about compliance, making sure that you followed the former. Now, under the International Treaty for Plant Genetic Resources for Food and Agriculture, as well as the PIP framework under WHO, that number two is not true, but we focused last week, and for a biodiversity-focused community, assume for yourselves that 2 is your rule. For number 3, every country can design their national laws and their ABS frameworks in their own way, and that information, the first place to start is the ABS clearinghouse. Now that's a very, very, very short summary of Scarlett's 45 minutes, but that's what you need to know of the sort of status quo until we get to 2016. Another sort of backwards historical note that I wanted to make to introduce this topic to you is what we've learned as scientists about international negotiations. So when the Nagoya Protocol was negotiated in Nagoya, Japan, in the province of Aichi, there was a horse trade, or in German we call it a Kuhhandel, the trading of cows instead of horses for whatever reason. And put very simply, there is always a tension in negotiations of saying this deal has to be made and this deal has to pay for it. So in some very oversimplified ways, the importance of creating the Aichi targets, which set the strategic plan for biodiversity from 2010 to 2020, was the trade with pushing through the Nagoya Protocol on access to genetic resources. And that's a really important moment to realize that there will be sort of landmark negotiations that require politicians and negotiators to make sure that they see what's happening in the other lane and the likelihood that they will need to make a deal, to cut a deal, even if their stakeholders aren't crazy about it at the time. So imagine Scarlett's talk. Imagine if you were sitting there 2 weeks ago and you thought, man, all of this effort just to get access to physical biological samples, what if I just went to the open public DNA databases and used some tools from synthetic biology and got to skip the hassle of the Nagoya Protocol of access and benefit sharing? Maybe we don't have to always do field sampling. Maybe we can surpass or bypass those national laws. And that sort of whisper, I guess, if you will, was what kicked off what we're going to call, for simplicity, the DSI debate. This tension between wondering if ABS laws could be avoided because of open access public DNA databases. So from 2016 to 2020, I've sort of titled this section of the talk as the emergence of the debate on DSI under the CBD. So the question of DSI actually emerged, as I just sort of hinted at, from a negotiation track on synthetic biology, the implications of synthetic biology for the Convention on Biological Diversity, and related topics into the Cartagena Protocol on biosafety. We don't have time to get into those, but it was brought up by negotiators that potentially benefits from digital sequence information should be in the scope of the CBD and the Nagoya Protocol. There were then additional negotiations, a study that was done, and this topic was pushed on to COP14 and attached to the open-ended working group to build and to draft the post-2020 Global Biodiversity Framework, which is the biodiversity framework or the strategic plan for biodiversity from 2020 to 2030 and actually beyond. And then moving forward to COP15, the need to adopt that GBF package deal. So to zoom in a little bit more on this, Fabian Roden and I wrote a policy piece several years ago that kind of mapped out the various meetings. So you can see at the top at COP13, the topic gets introduced. There is an ad hoc technical expert group, the ADTEC, that meets in 2018. GGBN sent a representative, their, I think, director or chair of the executive committee at the time, Dr. Ole Seberg from Denmark. And then their recommendations went forward on to SUBSTA, which is yet another acronym for the subsidiary body on scientific, technical, and technological advice. And in parallel, there was a study done by Rachel Weinberg and Sarah Laird that was a sort of fact-finding and scoping study on DSI. Because this kind of kicked off and the AgTech was very controversial and was not able to find consensus to come up with recommendations, at COP14 it was still very heated. And so the decision to COP14 basically said, let's do another AgTech and let's pass on our recommendations of the AgTech onto the open-ended working group. Now, this might not look at all interesting, these little blue boxes. What is super interesting is to note the alphabet soup change here between SBSTA and OEWG3. This means that you have now tied the DSI issue to the same negotiation body, the open-ended working group, that is negotiating the Global Biodiversity Framework, meaning that idea of forecasting a horse trade is inherent because DSI is not reporting to the technical body, the SBSTA, it's now reporting into the broader Global Biodiversity Framework. We'll see the implications of that towards the end of the talk. So after COP14, because of the decision at COP14, there were potentially 4 studies that were commissioned. So one on the concept and scope of DSI, one on databases and traceability, and then a 4th on domestic measures that had already begun on DSI. So we're going to zoom in on this, and what's really interesting here, having the GGBN as our host for today's webinar, is that here's the document that I just mentioned that commissioned those studies, and here is the text. It is very hard to know sometimes in UN fora how important text will be, but the one that we're going to zoom in is on this letter C and D, a peer-reviewed study on ongoing developments in the field of traceability and on public and private databases. So together with Katie, as well as a number of other colleagues, we decided as members of the Global Genome Biodiversity Network to respond to a call for proposals to do this study. And in that call, it had indicated that an application that included both a study on DSI traceability as well as on public and private databases would be highly welcome. The Secretariat, because it's very unusual to commission 4 studies, was interested in sort of economies of scale and strongly encouraged applicants to merge those together. That study is available at the following link, but what I wanted to do today is just to give you kind of a high-level view of the major take-home messages that the colleagues at the study group from GGBN put together. Again, our terms of reference were quite clear. We were supposed to include information on developments in the field of traceability, including how it is addressed by databases, and how and whether public and private databases make their data available, the size of them, the numbers of them, et cetera. And so we kind of had a very broad remit and about 6 months to put this study together. It kind of ruined my summer 2019, if I remember. So the major take-home messages that we sent to parties is that the International Nucleotide Sequence Database Collaboration, the INSDC, is, for the scientific community, the core infrastructure for holding nucleotide sequence data, which policymakers call digital sequence information. So we focus really on the data type of DNA and RNA, but of course, the policy word of choice is digital sequence information. We also pointed out that while there is this sort of exchange and mirroring of data between the 3 members of INSDC, the NCBI, EBI, and DDBJ, there are lots of other tools and services that each of them offer that are different and complementary to that global DNA data set. And that enables a huge amount of public good to come out of these interdependent tools and scientific portals. We also noted some of the finances. We did a little bit of digging through documents and came up with a very rough estimate that this infrastructure to run it across all 3 large databases costs about $50 million per year. We noted the data is synced and it is currently free to all users and does not require registration. We also cited the science article that the INSDC members had published in 2002 that notes a uniform policy of free and unrestricted access. that it will not attach statements to records that restrict access to data, including no restrictions on or licensing requirements, that the database records submitted to the INSDC will remain permanently accessible and will be fully disclosed to the public. Just to sort of give them the sense of where the DSI scientific community was starting from. And we also noted, because our terms of reference asked how do other databases, what kinds of terms of use do they have, that many other downstream or interconnected databases to the INSDC essentially inherit these terms and conditions by pulling their data out of the core or main database. We also thought it was important to note why scientists put their DSI or their nucleotide sequence data in a public database for everyone to see and use. This isn't because we're trying to show off per se, but rather because the journals in which we're publishing our scientific results will not publish papers unless they see the data in a public archive. And that for sequence data is almost always the INSDC members. That our grants that we receive to do public research, that those agencies that give us the money very often also have open data policies that require it. Another good reason is that the ethics of doing science correctly mean that we have to enable other scientists to reproduce our work. And there is a sense of reproducibility, of integrity, and also, of course, of data security. You know, if your lab burns down, if you put it into the global archive, the data is not all lost. And finally, another important message for policymakers is explaining that letters by themselves, that single pieces of DNA, don't actually mean anything until they have context. And that context is enabled only once they've been able to be compared to the global whole. We also did some fun world maps and showed that there are about 10 to 15 million total users of the INSDC. So these are not publicly available figures. We received these directly upon request, by request to the databases. And you can see the most users are in the United States, followed by China, India, Japan, etc. If you take that $50 million running cost, that means that there are about 50% of users that live in countries whose governments do not contribute to the running infrastructure costs, meaning in some ways, this is a global non-monetary benefit that enables access to this global archive. We also looked into the country metadata fields for DNA records in these databases. So there's a field that is called /country, although I think it now is called /geographical origin because there are because of the High Seas Treaty, basically. And you can find out through that metadata field where the original genetic resource, where the physical sample came from for these sequence data. And in fact, they come from every country in the world, and the major providers of access to genetic resources are, again, very similar countries as on the previous slide. Although you do see some additional countries here, like Mexico and Brazil, that you did not see as much or in the same ranking on the previous slide. Although these data are somewhat limited because only 16% of the total global data set actually has country-associated information, so they should be enjoyed with caution. Too far. That study was delivered to the AdTech right at the beginning of the pandemic. So in March 2020, it was like one of the first online meetings that I ended up doing in the pandemic. And in parallel, we had a new project funded by the German Ministry for Research and Training that was called Wild DSI. And we had done a lot of work and research to try to develop policy options for this DSI debate, recognizing that the study that I just explained wasn't going to be quite enough, because ultimately the pressure to get a deal on the global biodiversity framework was going to mean that policymakers were not going to leave DSI alone, but rather we're going to be looking for solutions. And so we published this white paper in October of 2020. There's a YouTube link if you want to go back and watch the hour-and-a-half webinar if you're not tired of webinars yet. And this was also done in close partnership with the Leibniz Institute in Gotteshaben for, I forgot the German translation, for crop resources, basically the plant gene bank here in Germany. Sorry, Jens, if you're watching this. So the Wild DSI white paper really focused first on asking scientists how to maintain open access and yet still engage with benefit sharing. And we collected a variety of case studies of why and how open sequence data contributes to the public good. I don't have time to get into that, but there were lots of really interesting examples that came out of the project. In the white paper, we really outlined and said, from a scientific perspective, here's our top 5 wish list. We want open access, we want simplicity, we would like you policymakers to learn from the Nagoya Protocol and the complexity for compliance that has arisen out of that. We would like a future-proof solution and a legal certainty in using sequence data. And also, because of the complexity of the Nagoya Protocol, if and where possible, opt-in options for physical genetic resources. We presented 5 open access policy options where we said these are options that would still enable openness to the data and generate benefit sharing. I won't get into these now, but what was really encouraging is that several months later, as parties indeed began to look for policy options, we were able to find these options in this rainbow chart that became one of the most popular graphics in about a year of webinars that continued during the pandemic sort of lockdown. So in March 2021, these policy options were presented by the CBD Secretariat, and we were very fortunate that a lot of the ideas that we had presented in the white paper were taken up into this sort of international standard at the time. And what you can clearly see here with the green colors on the right is that at this point in the negotiations, there are definitely bilateral mechanisms that are being considered, the darker green, versus there are more multilateral mechanisms. And this sort of gives us the option, again, to sort of help and to steer policymakers to understand what is lost for science, the more that we move towards the darker green colors. Again, there have been webinars on this chart and the implications and lots of informal and formal workshops on this. We don't have time to get into this, but the point here of this slide is to show that if the scientific community is prepared in time, you actually can have your ideas taken up and reflected in the broader policy process. We also used this time period in the online discussions that were happening during the pandemic to acknowledge the important arguments that were being raised, and particularly by low and middle income countries, that the reason that there was a call for benefit sharing is that in fact there has been an objectively an explosion in sequencing data since the Nagoya Protocol. So you can see here the Nagoya Protocol is adopted in 2010. And if you look at the exponential growth over time, and in fact, NCBI doesn't even graph the data this way, they graph it in logarithmic terms because the data grows so fast. And if you look at the tools that are available to biology through synthetic biology, through CRISPR-Cas9, they've also changed a lot since 2010. So here's just a table from a paper that shows where there's more stars, we're better at doing these kinds of technologies. Meaning that if you take big amounts of open data, really great biotech tools, smush them together, there really can be a gap in receiving benefits from the data that arose from your biological or genetic resources that came from your sovereign country. We also noted that there is a huge need in science for interoperability. And this is something that's really difficult to explain to policymakers that are not familiar with the daily practice of biology or bioinformatics. So this is a great graph from colleagues mainly based at EMBL EBI, but associated with the Elixir core data resources, showing just how much data interconnectivity there is between just these 19 databases. And actually, this happens on an even larger scale that can't very easily be graphed for hundreds of millions of sequences, for thousands of databases, and for hundreds of data types. So we worked really hard to sort of find images that could say, like, we're not calling for open access because it only is about the INSCC. It's also about the interconnectivity of being able to zoom in certain scientific fields and transform data into other data types, into other data collections or mixtures. And a colleague here at the DSMZ, Julia Koblitz, has also been thinking a lot about that same concept, but with a slightly different, more biological map. that understanding how biology works requires an integrative thinking, which ultimately, of course, requires integrating the data. So you can't just look at a single, nucleotide sequence data over here and be happy. You have to also look at lots of other things that are arrows farther down in order to answer your scientific questions. And then on the question of future-proofing, we were also noting that while everybody was focused very much in the negotiations on the sequence data, on ACs, Gs, and Ts, that there was also going to be data growth in other areas. So if you zoom in right now on that black line for macromolecular structures, you can see here on this now exponential graph, where the sequence data is in red and things like protein structures are in black. There's not huge amounts of change at this point. However, you're going to have breakthroughs in technology. So if you look, for example, oh, sorry, this is a slide from another thing. If you look at what's coming, there are going to be revolutions, and many of which might come out of artificial intelligence, that will rely on the use of the global data set or the collective aggregate. So you can see here the headlines from the application of the DeepMind technology to crack a 50-year-old problem, increasing those 1 million roughly protein structures that were in this graph up to 200 million basically overnight, right? And that breakthrough means that designing a good benefit sharing mechanism that can deliver fairly and equitably benefits to parties needs to sort of look into the future and not think about individual sequences, individual data records, but much more about the overall trends and direction of travel of where science is going, which means for designing the DSI benefit sharing mechanism, the more that you can get towards holistic approaches, the more likely the mechanism will deliver. Whoops, went too far. The other thing we were doing around this time period was trying to deliver empirical data to test the DSI policy hypothesis. So in the policy rhetoric at the time, many countries were sort of saying or thinking that DSI use was a one-way street, that developing countries provide access to biodiversity, that developed country scientists use and profit from the biodiversity, and we call this in the paper the provider-user dichotomy. It sounds a little bit fancy, but basically this one-way street. In order to test our hypothesis, or my personal belief, that the use of DSI is actually highly interdependent and highly integrative, that scientists everywhere use everything, and it really just depends on the scientific question that they're asking. So we used real data from open scientific publications. We looked at 9 million sequences that had country of origin information and 118,000 open access scientific publications, which cited those 9 million sequences. So how we did this was we took the publications and we did some text mining. So you would take a publication like this, you would see Mr. Christopher Cruz, you would see he was physically located in Peru, and so that would make him as the geographical affiliation in Peru, and then you would take, through text mining, all of the different accession numbers indicating a sequence record from the publication, look them up using automated methods in one of the INSTC databases, and then ask about the country information, in this case, Brazil. So if you took this particular publication, it would give you one count for providing country is Brazil, and because that's where the sequence came from which the original genetic resource was sampled from, and the using country is then Peru in this country, where the author that published the publication citing the DSI was. If you do this across all of that data, you can generate figures like these here for Colombia. On the left, you can see that Colombian scientists are using DSI that has been sourced from 75 different countries, and those countries are sometimes in Latin America nearby, and sometimes they're far away. And then you can also see that Colombian scientists are providing DSI that is used by 56 other countries, meaning that this relationship between providing and using doesn't necessarily fall along the predictions made by the one-way street hypothesis, but rather this speaks to that interdependency that we also thought was likely. If you take those kinds of graphs and you graph one point for every single country, where on the x-axis you have scientists that are using the DSI, and on the y-axis providing access, you can see that the relationship between providing and using is actually sort of tied in a general trend line. And perhaps most importantly, we could talk again for a while about this graph and talk about individual countries, but in general, there is no country that is only providing access to DSI, that there are no dots up there. And similarly, there are also no countries that are only using. This sort of brings home the traditional, I think, very sciencey message that all countries are users and all countries are providers. Another way to graph these data is to look at the country of origin for the genetic resource that gave rise to the DSI, which is on the X, and then group those data by the traditional UN groups. So Eastern European group in green, the African group in yellow, the red for Latin America, Western Europe and others, purple, and Asia Pacific for blue. So if you zoom in, for example, on the African group on the far left, you can see the biggest users of African sourced DSI are in fact yellow, the African group users themselves, meaning that scientists use local data more than foreign data. And this is incredibly powerful because this trend is true for every single column in this bar graph. However, what is also quite striking is the amount of publications that are coming from those DSI records. There in Africa, in Eastern Europe, and in Latin America, there is a true capacity gap for being able to harness and use these data. And it was really thinking and reflecting on this sort of interdependency that was one of the motivations for, at the time, a group of about 15 or 20 of us, again, international, and a lot of the traditional suspects that we had gotten to know through the GGBN study, as well as others, to found the DSI scientific network and to really conduct international outreach. So we founded this originally in 2020. We now have over 80 members from 28 countries from every regional group. We have online webinars that we conduct to raise awareness. We try to always send a delegation of 2 to up to 10 people to different CBD meetings. We conduct bilateral briefings, develop position papers, and conduct coordinated outreach amongst ourselves. And there's a website there. I wanted to just give you, I hope I'm doing okay on time, Rob and Katie, to give you a kind of a flash through of some of the outputs of the DSI scientific network. So we published in advance of the resumption of in-person physical negotiations in Geneva in 2022, an open letter, again, calling for open and multilateral. Then we were able to generate over 1,000 individual signatures, as well as a number of diverse and also high-profile institutional signatures to this letter, to really say this is not just the position of 10 people, this is really a broader international call for this openness. And importantly here, and why we thought this was an important compromise position compared to previous scientific engagement on the topic, was not to take a blocking position and say, we don't want you to make any rules for DSI. We see the need, in fact, for benefit sharing, that is an important step for fair and equitable benefit sharing for the foundations of the CBD and its founding objectives. However, we do feel that there is a better way to do this than other options that are on the table. And that was this open letter. Then we also took a little bit of a deeper dive amongst ourselves within the network to put our ideas of what that could look like into a more concrete policy proposal that came out in Nature Communications around the same time, to say that multilateral benefit sharing is the way to go, and that it can support both the science as well as biodiversity conservation itself in the context of the upcoming Global Biodiversity Framework. In that paper, you'll see some overlap with the Wild DSI white paper from 2020, but it does sort of evolve. We call it for open access, no surprise there. And there we sort of introduced a new concept called decoupling, meaning that the traditional benefit sharing acronym has been ABS, and people say it all in one breath. as if that those letters must always stay together. And instead we were saying here, you know, open access is possible and there could be a divorce from benefit sharing. And by divorce, meaning these two things don't have to be coupled. You don't have to control access in order to enable benefit sharing. And that's where this word decoupling kind of comes into play. It is possible to have openness in the access front and still enable benefit sharing. Simplicity, we pointed out with, again, the statistics that I kind of showed you about the sizes of the data set, about the data exchanges, that the science is complicated, and therefore that the legal framework certainly can't be. The need to harmonize, this hasn't come up yet in this talk so far, but there are another number of other policy-making decisions happening in other UN fora, and the complications for accessing genetic resources would make a complicated DSI solution even more difficult to implement. We also see the need, because, and especially this community must understand this, that DSI itself is a driver and a tool in the biodiversity crisis for conservation and sustainable use, and also for the development of the bioeconomy for green growth, that there is an interconnectedness here. So if you create a DSI solution that doesn't work very well, you'll inherently also hamper the ability to deliver on the other objectives of the CBD. And finally, we called for the fairness and the transparency that public DSI databases need to deliver to provide, for example, data for GBN indicators, potentially for reports on countries of origin, but we insisted that this cannot be a bilateral or a control-based Nagoya-like mechanism, because there were, for example, at the time, policy proposals that had called for databases sort of acting like the benefit sharing police, where they would register users, they would check on users, and then they would report on those users to the benefit sharing mechanism. And we pointed out this is not the remit or the job of database managers. We also contributed at the time, and there are lots of other things that I could explain, but I found this one particularly compelling, case studies on examples of how DSI is used. So we looked, for example, at the patent that Moderna filed even before it was declared a public health emergency of international concern, and pointed out to policymakers and negotiators that in fact there were no SARS-CoV-2 sequences in this patent. The idea that you could predict what kind of data commercial or non-commercial users would use and be able to track and trace it into the benefit sharing equation would be very difficult, in fact. We noted this was a method patent, and that there were actually 150 sequences from public databases from many different families. There were 96 new sequences that were generated by Moderna, and that these were from dozens of different countries, as you can see in the table up there. This was developed into a case study, as you can see here, and we really tried to point out in this case study that in order to do modern biotech, and of course at the time the pandemic was on everybody's minds, you need to take many natural sequences to understand a very specific thing, in this case, the spike protein, and then use that integrated scientific knowledge to create a novel product. And in this case, there was only 70% identity to a natural sequence, and we pointed out if the benefit sharing mechanism is based upon benefit sharing only from individual sequences, you won't get benefits from a COVID-19 vaccine, for example, because it would be easy to sort of create an artificial sequence that wouldn't fall under the tracking and tracing kinds of mechanisms. So what happened to COP15 then, after all this hard work, after all of these scientific outreach, scientific communication, policy engagement, et cetera? The question that we all had in flying to Montreal was, will parties be able to make a deal on the Global Biodiversity Framework and a deal on digital sequence information? So there were actually 2.5 weeks of negotiation. As always, they went very late into the night, into the last days, and parties were barely able to meet the time pressures that they had outlined for themselves. And for our part, the DSI Scientific Network took our multilateral open access message to COP. So we had a side event, for example, colleagues there on the left from Brazil, from India, and Jens and I from Germany, and a broader delegation from many different countries to do outreach there on the floor at COP. And finally, at the end of those 2.5 weeks, I think around 4:15 a.m., the gavel fell. and a deal was made. And for most of us in the room, we didn't quite know what was going to be in the deal. And the reason is because in those last 2 days of the meetings, it becomes a high-level ministerial segment, and a lot of the deals are made behind closed doors. So you don't see the text until the very last minute because it's being negotiated by the ministers at that point. So what ended up being in it, it was in fact a horse trade or a Kuhhandel, again, just like the Nagoya Protocol and the Aichi targets. So this is the new global strategic plan for the Convention on Biological Diversity. It is called the Kunming Global Biodiversity Framework. And it is a take it or leave it package of 5 items that was introduced by the Chinese presidency. So what happens is they basically take the other items that are being negotiated. So I think there's about, I don't know, 15 to 20 items, and they get brought down as L documents. And this is the package that negotiators are told, you either take it or leave it. You can either take all of this or none of this, but there is no more tweaking with individual things. And as you can see, number 5, DSI, the focus of this webinar, was part of that package, meaning at some point, policymakers' hands are tied. They either have to choose between taking the GBF and the monitoring framework and the capacity building plan, as well as the finances, or not, but they can't just say no to DSI, right? It's part of the broader package, and that's the way that you get big deals through. What was in the decision then? It was a list of principles that define the parameters or the criteria for the development of a DSI benefit sharing mechanism. It does have a clear focus on open access, combined with improvements in data governance and equity. It commissions 3 studies, and the most important thing is it decides to establish a multilateral benefit sharing mechanism. Diving in a little deeper into that, the core principles here, and I've highlighted in bold the words that mean the most to me: to provide certainty and legal clarity for providers and users, to not hinder research and innovation, to be consistent with open access to data, and mutually supportive of other access and benefit sharing instruments. However, so that's the good news. The good news is the principles. The sort of neutral news, before you breathe a big sigh of relief, is that the COP decision is also everything and nothing. In some ways, it is incredibly important that paragraph 16 says, decides to establish, as part of the Global Biodiversity Framework, a multilateral mechanism for benefit sharing from the use of DSI on genetic resources, including a global fund. It also decides to establish a fair, transparent, inclusive, participatory, and time-bound process to further develop and operationalize the mechanism. And that's what's happening right now. Ultimately, we find ourselves in this world where the DSI benefit sharing, the path the CBD is taking is more complex than we could discuss today. So we focus very much in here on the CBD as well as the INSCC databases, less on the Nagoya Protocol. But the green circle there, the International Treaty for Plant Genetic Resources for Food and Agriculture, all of the plants that feed the world, we haven't talked about. There is also a new high seas treaty known as BBNJ, which is biodiversity beyond national jurisdictions, and another meeting in which negotiators will meet again next week under the WHO, the CA+ agreement, which is a pandemic agreement that will create rules and regulations for pathogens, as well as the existing WHO pandemic influenza preparedness framework. So suffice it to say, there is a lot of complexity. There are also, as Scarlett mentioned, countries that have developed national DSI rules, and there are also private databases that sort of, we don't really know how and if and whether they will be involved in this broader DSI benefit sharing debate. So we, despite the roughly 40 minutes of me talking at you, there's a lot more complexity here that we couldn't cover. The negotiations are basically non-stop right now, and while I focus on the CBD, I've spent a lot of time in recent months thinking about the WHO work, and before that, the BBNJ process. But you can see that a lot of things are coming up towards the end of the year, and in fact, at the World Health Assembly in May even, and likely also beyond. And because of all of that, the effort that the DSI Scientific Network is really focused on, and has written a publication that hasn't yet been accepted or published, But we're thinking a lot about what we're calling DSI harmonization and the fact that different United Nations fora are basically on a DSI collision course, and the need to, on the one hand, of course, do benefit sharing across all of these fora, but the recognition that if those rules are not somehow harmonized or complementary to each other, that when you ask a question, for example, about green plants, you will have multiple different fora and their rules on DSI benefit sharing that you would have to address and handle. And so again, trying to be ahead of the curve of where policymakers are going to provide advice now, not only to the CBD, but also to other fora, and to attempt to get some kinds of harmonization. I will note here a joke, a lawyer joke, that when lawyers talk about this, they tend to say regime fragmentation, so more like the negative thing, like when rules aren't compatible with each other, it's regime fragmentation. So the scientists are calling for harmonization to avoid the lawyer name for it, regime fragmentation. And a final piece, just to kind of have on your radar, a relatively new publication that ultimately I believe that the DSI debate, and again I wanted to close the full circle back to Scarlett's talk, if the DSI benefit sharing mechanism works, if it delivers real tangible benefits, it ultimately could maybe enable the long-term redesign of how access and benefit sharing is done. And here in this paper, we talk about 4 principles and provide, again, recommendations to policymakers on how that could be done. This is a bit more of a philosophical piece, but I do see the DSI discussion as a chance to relook at how we can do fair and equitable benefit sharing better. Just to wrap up for you, what I hope you heard today, the very last slide, is there is agreement now, since 2022, amongst the parties to the CBD, which has near universal ratification, except for the United States and the Vatican, that benefits from digital sequence information shall, as it must, be shared under a multilateral mechanism. However, that mechanism is not yet in place. There is no international rule yet that we can point to say it's going to be like this. And in fact, the next 2 and maybe beyond years will be critical to continue to bring forth this message of open and multilateral. And the real open question and the risk for this community is whether or not scientists can continue to bring that message forward. I hope you've also noted that there is a relatively small group of scientists working on a very small budget to engage in this policy decision-making process. And so that's a final open door remark, that if you want to be part of this work in any way, please feel free to reach out to us at the email there below. And with that, Rob and Katie, I hope you've gotten enough of a crash course on DSI, and I look forward to our discussion.
Thanks, Amber.